Part 5. Modern Fluid Mechanics at the Scale of Life. The new scale of fluid mechanics. Fluid mechanics has moved far beyond the familiar images of water in pipes, air over wings, and pumps driving liquid through industrial equipment. Those classical problems remain essential, but one of the field's great modern advances has been the creation of tiny, controlled environments in which fluids can carry out work that once required an entire laboratory. At the microscale, flow is not merely a smaller version of what we observe in rivers or pipelines. The same conservation laws still apply, but reducing the length scale changes the relative importance of the physical effects. Inertia often retreats, while viscosity, diffusion, capillarity, and interactions with surfaces become increasingly influential. A microchannel may carry fluid gently and predictably, yet within that apparently calm motion, it can separate cells, create droplets, maintain a chemical gradient, expose tissue to controlled shear, or deliver reagents in a precisely timed sequence. This change of scale can be heard in the Reynolds number. Because the characteristic length is small, many microfluidic systems operate at low Reynolds number, where viscous effects dominate over inertia and flow is generally laminar. That orderliness is not a limitation alone. It is one of the reasons microscale flow can be designed with such precision. Microfluidics is generally understood as the science and technology of manipulating very small quantities of fluid in channels, with dimensions typically measured in tens or hundreds of micrometers. Its importance comes not from smallness by itself, but from integration. Fluid control can be combined with chemistry, biology, optical detection, electronics, sensors, and materials inside a compact device. The channel is no longer merely a conduit. It can become a reactor, separator, incubator, diagnostic pathway, tissue environment, or manufacturing chamber. A laboratory becomes a chip. The ambition to miniaturize chemical analysis emerged before the terms organ-on-chip and personalized medicine became familiar. In 1990, the analytical chemist Andreas Manz and his colleagues Nathaniel Graber and H. Michael Widmer, working in the corporate analytical research laboratories of Ciba Geigy in Basel, Switzerland, proposed a miniaturized total chemical analysis system. The idea did not come from a university at all. It came from an industrial laboratory that needed better ways to measure what its chemistry was doing. Their proposal was more ambitious than shrinking one instrument. They imagined integrating a sequence of analytical operations on a small platform, using fabrication methods related to those developed for microelectronics. That proposal helped give the field both a direction and a vocabulary. The phrase micro total analysis system, commonly abbreviated as micro-TAS, captured a new engineering goal. A sample would not simply pass through a small tube. It would encounter a coordinated sequence of operations. Transport, mixing, reaction, separation, and detection could become parts of a single fluidic architecture. The concept was powerful, but a concept still needed a practical way to build devices. Early microsystems drew heavily on silicon and glass fabrication. These materials offered precision, but the methods could be demanding and expensive. A major change came when George Whitesides and his colleagues helped make soft lithography widely accessible. Whitesides, a Harvard chemist and one of the most widely cited chemists of his generation, led a group that trained several hundred graduate students and postdoctoral researchers. Many carried these methods into chemistry, biology, engineering, and materials science. Using soft lithography, researchers could reproduce intricate channel networks in transparent elastomers, especially polydimethylsiloxane, commonly known as PDMS. The significance of soft lithography was not only technical, it was also cultural. A chemist, biologist, physicist, or engineer could design a chip around a scientific question without first becoming a specialist in semiconductor manufacturing. Transparent devices could be bonded to surfaces, connected to tubing, and observed through a microscope. Microfluidics became a meeting place for disciplines that had previously approached small-scale transport from different directions. Whitesides later asked an uncomfortable question about the field he helped shape. If microfluidics offered small sample volumes, rapid analysis, compact devices, and precise control, why had laboratories and clinics not already filled with labs on chips? The question remains useful. A beautiful flow pattern in a research device is not yet a reliable product. Packaging, pumps, materials, calibration, manufacturing, and ease of use can determine whether an elegant idea ever leaves the laboratory. Laminar flow, diffusion, and the art of controlled contact. Imagine two streams entering a microchannel side by side. One carries a dissolved dye, and the other is clear. At the confluence, the streams do not immediately dissolve into turbulent disorder. They can travel together with a visible boundary between them. The liquids are not refusing to mix. They are mixing mainly by molecular diffusion across the interface, because turbulent eddies are absent. Diffusion becomes effective over short distances, but its characteristic time grows with the square of the distance. If the diffusion distance is halved, the corresponding time scale can fall by roughly a factor of 4. This scaling explains why miniaturization can accelerate some transport processes even when the flow itself remains calm. It also explains why a channel that is only modestly wider may mix much more slowly than expected. This creates a choice for the designer. Sometimes rapid mixing is needed. In that case, serpentine channels, repeated splitting and recombination, obstacles, hydrodynamic focusing, acoustic forcing, electrokinetic motion, or segmented flow can shorten diffusion paths or continually rearrange interfaces. At other times, mixing must be restrained. Stable laminar streams can create controlled concentration gradients, expose opposite sides of a tissue to different conditions, or allow the movement of a molecule from one stream to another to be measured with spatial precision. The absence of turbulence, therefore, does not make microscale flow passive. It makes geometry more important. Every bend, junction, constriction, surface treatment, and interface becomes part of the experiment. Modern microfluidics is classical fluid mechanics made intimate. The walls are always close. Diffusion competes with advection over observable distances, and the interface is often an active element of the design. Droplets as miniature laboratories. At the beginning of the 21st century, researchers at the California Institute of Technology, including Todd Thorson, Richard Roberts, Francis Arnold, and Stephen Quake, demonstrated how the meeting of two immiscible fluids inside a micro device could generate remarkably regular droplets and more complex ordered patterns. Their work showed that low Reynolds number flow need not be featureless. Interfacial tension, pressure, channel geometry, and viscous stresses could interact to create organized structures far from equilibrium. This helped open one of the most elegant branches of modern fluid mechanics, droplet-microfluidics. Instead of treating a fluid as one continuous stream, the device divides it into a sequence of tiny compartments. Each droplet can carry a cell, an enzyme, a strand of genetic material, a drug candidate, or a chemical reaction. Long sequences of droplets can pass through a device without constructing an equivalent number of separate vessels. Droplet formation is a negotiation among viscous stress, interfacial tension, wetting, geometry, and flow rate. In a T-junction, one phase enters from the side and interrupts another. In flow focusing, surrounding streams squeeze a central stream through a narrow region. In coaxial arrangements, one fluid enters within another. By changing fluid properties and operating conditions, the designer can tune droplet size and production frequency. Once formed, droplets can be incubated, sorted, merged, split, analyzed, or solidified into particles, capsules, and microgels. This is why droplet-microfluidics has become valuable in high-throughput screening, biomolecular analysis, sequencing workflows, diagnostics, and single-cell studies. It is also closely connected to emulsion science, the same interfacial mechanics that govern foods, cosmetics, polymer blends, and petroleum emulsions are now used to construct miniature experimental worlds one droplet at a time. In food science, the same control produces emulsions, microgels, and microcapsules from food-compatible ingredients, placing a sensitive nutrient inside a protective structure whose interfacial behavior, coalescence, and release can then be studied directly. At this scale, an emulsion is no longer merely a mixture. It becomes an architecture for experiments. from channels to complete workflows. A lab on a chip extends microfluidics from fluid handling toward an integrated analytical process. Consider an illustrative journey. A small sample enters a disposable cartridge. A filter or geometrical barrier removes unwanted material. Flow carries the remaining sample toward a region where it meets a reagent. A short diffusion path permits molecular contact. A reaction changes an optical or electrochemical signal. A sensor records the change. The operator sees a result without manually transferring the sample among several instruments. Not every device contains all these operations, and integration is often the most difficult part. Nevertheless, the objective is clear. A workflow that once required pipettes, tubes, centrifuges, incubators, and detectors can be reorganized as a sequence of fluidic events. Smaller sample volumes may reduce reagent use, while compact systems can bring selected analyses nearer to clinics, field sites, farms, or manufacturing floors. Microfluidic diagnostics have been investigated for nucleic acid analysis, immunoassays, pathogen detection, cancer-related liquid biopsy, food safety, and environmental monitoring. The value is not simply portability. Precise control of transport can improve how a sample is prepared, how long it contacts a reagent, how strongly a target binds to a sensing surface, and how background material is removed. Yet the integrated device is only as reliable as its weakest stage. Chemical engineers will recognize the principle from reaction kinetics, where a sequence of steps proceeds no faster than its slowest one. The rate-limiting step governs the whole. I put it to students this way. On a crowded street, the traffic moves at the pace of the slowest cars, and the fast cars, however capable, gain nothing from their capability. A microfluidic workflow behaves the same way. A sensitive detector cannot compensate for poor sample preparation. A perfect channel geometry is of little use if bubbles block the flow. A compact cartridge is not truly accessible if it requires a complex external pump, fragile tubing, or expert calibration. The lab on a chip vision succeeds only when the entire workflow, not merely one striking demonstration, becomes dependable. Living cells under flow. Microfluidics becomes most vivid when the transported material is alive. A cell is not a passive tracer. It senses shear stress, confinement, the transported supply of oxygen and nutrients, neighboring cells, the stiffness of its surroundings, and the chemical signals carried by the fluid. An ordinary culture dish exposes cells to a comparatively static pool of medium. A microfluidic system can perfuse them continuously, impose gradients, arrange them in defined geometries, or expose them to a drug while their response is observed. Three-dimensional culture adds further realism. Cells in a spheroid, organoid, or tissue-like matrix experience diffusion limits, mechanical support, cell-to-cell communication, and gradients of nutrients and drugs. These features can make their behavior different from that of the same cells spread across a flat surface. Microfluidic perfusion does not recreate an entire body, but it can place a controlled transport environment around a selected biological structure. One recent example moves beyond observation toward manufacturing. Certain cancer therapies take a patient's own immune T cells and give them a new surface protein, a chimeric antigen receptor, that lets them recognize and attack tumor cells. Cells modified this way are called CAR T cells, and manufacturing them is slow, expensive, and difficult to do consistently. In 2024, Weixiang Xin and a multidisciplinary team working across Singapore and the Massachusetts Institute of Technology reported a compact, automated, closed-system microfluidic bioreactor for producing them. The system was used to activate human primary T cells, introduce the genetic instructions for the receptor, and expand the resulting cells under controlled perfusion. The study reported viable CAR T-cell populations and compared their functional properties with cells produced in a conventional gas-permeable culture well. The importance of this work is not that microfluidic CAR T-cell manufacturing has already become a universal standard. It has not. Its importance is that a small perfused chamber can be treated as a manufacturing environment, not merely as an analytical chip. Miniaturization can intensify control over nutrients, waste removal, cell density, and the local culture conditions, while supporting a complex multi-step biological process. The lung that breathed on a chip. One of the field's most memorable demonstrations began with a question about the lung. Could a device reproduce not the entire organ, but one of its most important functional boundaries, the delicate interface between air in the alveoli and blood in the surrounding capillaries? Dongjun He and colleagues in Donald Ingber's group at the Wyss Institute for Biologically Inspired Engineering at Harvard University answered that question with a lung on a chip reported in 2010. The team placed human lung cells and blood vessel cells on opposite sides of a flexible, porous membrane. Air occupied one channel, while culture medium flowed through a neighboring channel representing the vascular side. Cyclic mechanical stretching reproduced an essential mechanical feature of breathing. The point was not to claim that a complete lung had been miniaturized. The device reconstructed selected features of the alveolar capillary interface and allowed them to be studied under controlled conditions. The researchers examined responses to bacteria, inflammatory signals, and nanoparticles, and investigated how cyclic strain influenced those responses. This work reveals why fluid mechanics is central to organ-on-chip technology. Many organ functions depend on perfusion, transport, pressure, shear, deformation, and fluid-mediated signaling. Blood flows through vessels. Air moves through lungs. Filtrate passes through kidney structures. Interstitial fluid moves through tissues. Cells respond not only to chemicals, but also to the forces and transport rates around them. Organ chips have since been developed to represent selected features of lungs, intestines, livers, kidneys, the blood-brain barrier, tumors, and other tissues. They can support disease modeling, toxicology, drug testing, and studies of human physiology. Their promise is considerable, but so are their constraints. A chip must be reproducible, manufacturable, biologically relevant, and compatible with meaningful decisions. If it is too simple, it may omit essential biology. If it is too complex, it may become difficult to operate, compare, validate, or regulate. The most useful question is therefore not, can an entire organ be built on a chip? It is, which features of this organ must be reproduced to answer the question at hand? The goal is not maximal complexity. It is sufficient physiological meaning under controlled conditions. Reading, one cell at a time. Modern biotechnology depends not only on growing cells, but also on reading them. A population that appears uniform in a bulk measurement may contain rare and consequential differences. A tumor can include a small drug-resistant subpopulation, an immune cell sample a few strongly responding clones. Microfluidic systems can focus, separate, trap, encapsulate, culture, or lyse individual cells, and by handling them one at a time, they make that heterogeneity visible rather than averaging it away. Droplets can isolate single cells with reagents. Channel networks can guide selected cells toward different analyses. Integrated systems can connect cell handling with genomic, proteomic, or metabolic measurements. Blood provides a particularly demanding example. It is a concentrated suspension of deformable cells, platelets, proteins, and plasma moving through vessels whose dimensions vary enormously. In small vessels, the size and flexibility of the cells matter directly. Microfluidic devices can reproduce selected constrictions, shear gradients, adhesion conditions, and separation processes. These devices support research on microcirculation, clotting, cell deformation, cancer cell transport, and diagnostic isolation. The strength of these platforms lies in control, but their weakness can be the complexity of real samples. Patient-derived material is scarce, variable, fragile, and biologically heterogeneous. Proteins adsorb to surfaces. Cells adhere where they were not expected to. Channels clog. A design that works with a clean laboratory suspension may fail when confronted with blood, mucus, tissue fragments, or environmental debris. Life does not always behave like a calibrated solution. Programmable droplets, sound, and electric fields. Microfluidics has also moved beyond fixed pressure-driven channels. In digital microfluidics, discrete droplets are manipulated on patterned electrode arrays. Richard Fair and other researchers working on electrowetting helped develop the idea that fluidic operations could become programmable and reconfigurable. By applying voltage to selected electrodes, the device can change local wetting conditions and move, dispense, merge, or mix droplets. The analogy with digital electronics is useful, but should not be taken too literally. A droplet is not a bit of information. It has viscosity, surface tension, contact line dynamics, evaporation, contamination risks, and a chemical history. Still, addressable droplet control allows a device to perform different sequences on the same platform without assigning every operation to a permanently fixed channel. Other systems act on fluids without touching them at all. Acoustic fields can focus, sort, or pattern suspended cells and particles, while electrokinetic methods drive fluid motion or separate charged species. Nanofluidics extends the problem to even smaller dimensions, where confinement, surface charge, ion distributions, and molecular scale interactions become increasingly important. In such spaces, the wall is not merely a distant boundary. It may influence much of the fluid volume. These effects are used in studies of molecular transport, ion selectivity, filtration, DNA manipulation, and sensitive detection. AI as an assistant to fluidic design. Microfluidic experiments can generate large volumes of images and sensor data. A high-throughput droplet system may produce more observations than a person can assess manually. Machine learning methods are therefore being investigated for image classification, assay interpretation, flow control, design optimization, and the search for operating conditions. Artificial intelligence does not remove the need for physics. A model may identify a geometry associated with uniform droplets or classify cell responses in images. But fluid mechanics still explains how viscosity, interfacial tension, diffusion, wetting, and channel dimensions produce the observed behavior. Data-driven tools are most valuable when they work with physical understanding rather than conceal it. The same caution applies to automation. A controller can adjust pressure or voltage in response to a sensor signal, but a reliable system must still recognize bubbles, fouling, drift, biological variability, and conditions outside the data used to build the model. The future is therefore not AI instead of fluid mechanics. It is fluid mechanics joined to sensing, control, and carefully validated computation. Scientific and industrial reach. The reach of microfluidics is broad because many industries handle small quantities, sensitive materials, or complex mixtures. In pharmaceuticals, microscale systems support formulation studies, drug screening, controlled particle production, and analytical workflows. In diagnostics, they support selected point-of-care assays, nucleic acid tests, immunoassays, and cell or biomarker separation. In environmental work, they are being developed for field-oriented detection of chemical and biological contaminants in samples such as water. Food technology offers a revealing parallel. The same mechanics that help a biomedical engineer encapsulate a cell can help a food engineer encapsulate a nutrient, flavor, or sensitive bioactive ingredient. Chemical engineering also returns to the story in a new form. Small channels have high surface-to-volume ratios, short transport distances, and precise residence time control, which can intensify heat transfer, mass transfer, and selected reactions. Yet high performance in one channel does not automatically translate into high production. Industrial scale may require parallelization, robust distribution of flow among many channels, and manufacturing methods that maintain uniformity. Why elegant chips often struggle. Microfluidics has produced beautiful experiments, but translation remains difficult. Materials can absorb or adsorb molecules. Hydrophobic compounds may partition into polymers. Proteins may adhere to walls. Bubbles can interrupt flow. Cells and particles can clog narrow passages. Pumps, tubing, connectors, and detectors can make a supposedly compact device dependent on bulky external equipment. Variation in fabrication can change channel dimensions and surface properties. Sterilization can alter materials. Long-term cell culture demands stable temperature, gas exchange, nutrient delivery, and waste removal. A device intended for diagnosis must handle the sample as it actually arrives, not as the researcher wishes it would arrive. It must also be calibrated, packaged, transported, stored, and operated consistently. This is why Whitesides' earlier question still matters. The physics may be compelling, yet commercialization depends on the complete system. Reproducibility, standardization, manufacturability, regulatory acceptance, cost, and user behavior are not secondary details. They determine whether the technology becomes a scientific instrument, a useful product, or an elegant demonstration remembered mainly through a paper. Biological translation adds another layer. Cells vary from donor to donor. Tissues remodel. Immune components interact. A model can reproduce one key interface while omitting endocrine, neural, immune, or whole body effects. An organ chip is therefore not a miniature human. It is an engineered representation whose value depends on whether the selected features are appropriate for the question being asked. The goal is not to build the most complicated chip possible. It is to build the simplest system that preserves the physics and biology needed for a meaningful answer. Future directions. New materials may reduce dependence on the elastomers that made academic prototyping so successful. Thermoplastics, paper, hydrogels, and glass each offer different balances among cost, chemical compatibility, and manufacturability. No single material is ideal for every application. Automation will allow devices to respond to sensors and adjust flow conditions in real time, and machine learning may help researchers navigate large design spaces. But the decisive advances will still require understanding what changes when scale changes. The hierarchy among inertia, viscosity, diffusion, capillarity, surface charge, and biological response remains the foundation. Synthesis. From pipes to programmable biology. The story of modern fluid mechanics is the story of a field that became smaller, and by becoming smaller, became larger in impact. The principles that once helped engineers understand pipes, pumps, boundary layers, and transport now help them construct miniature environments in which cells can be cultured, droplets can become reactors, biological interfaces can be modeled, and molecular information can be measured. The people who shaped this development did not all pursue the same goal. Andreas Manz, Nathaniel Graber, and H. Michael Widmer imagined integrating chemical analysis. George Whitesides and his colleagues made device fabrication more accessible. Todd Thorson, Richard Roberts, Francis Arnold, Stephen Quake, and many others showed how interfaces could produce controlled compartments. Donghyun He and the lung on a chip team translated a living tissue boundary into an experiment under flow. Wei Xiangxian and collaborators showed how a microfluidic chamber could support a complex cell manufacturing process. Their work reveals a common pattern. Progress came not from making a conventional apparatus smaller for its own sake, but from asking what new control became possible when distances shortened, surfaces moved closer, and transport could be organized in space and time. In this new world, a channel is not merely a channel. It may be a reaction pathway, a blood vessel model, a tissue niche, a droplet generator, a diagnostic cartridge, a microbioreactor, a food encapsulation device, or an environmental sensor. Fluid mechanics remains the science of motion, but it has also become a science of controlled microenvironments and programmable biological interfaces. The broad view in this chapter was intentional. It shows the range of the platform, from laminar streams and droplets to cells, organ chips, sensing, and manufacturing. Breadth, however, does not reveal what the technology feels like when it is directed at one specific and consequential problem. For that, we turn to DNA analysis on a chip, which gathers the whole engineering chain into one story. Fluid must be moved without losing the sample. Molecules must encounter reagents. Temperature may need to be controlled. Surfaces must avoid unwanted adsorption. Signals must be detected and interpreted. The device succeeds only when physics, chemistry, fabrication, and workflow operate together.